Good day everyone, especially to Ma'am Alice Pahit. So today, I will discuss the different types of gene interactions. So what is gene interaction? So gene interaction is the process by which the expression of two or more genes influence one another in different ways as an organism develops a single characteristic. So the majority of the traits that comprise living beings are coordinated by various genes. Types of gene interactions. So gene interactions are divided into two categories. First, the allelic or non-epistatic gene interaction. So this gene interactions occurs between the alleles of single gene. Second is the non-allelic or epistatic gene interaction. So this gene interactions involves interaction between genes on identical or distinct chromosomes. So here we go. We're gonna talk about the allelic or non-epistatic interaction. So allelic or non-epistatic interaction has three dominants. This is the complete dominance, codominance, and incomplete. But before that, let's review the example of blood type and how someone with the same two alleles coding for the same trait would be called homozygous. And someone with different alleles would be called heterozygous. Again, it's called heterozygous. Also remember the concept of dominant and recessive alleles and how the A allele is dominant over the O allele in this example. This means that the same phenotype, blood type A, can result from these two different genotypes. So now, the example that I just gave you was an example of complex dominance. So if a person had a genotype AO, since our phenotype it's just blood A, it means that the A allele is completely over the O allele, and only the allele from the genotype is expressed in the phenotype. But there are actually three different patterns of dominance that I want you to be familiar with. And to explain this, I'm going to use a different example. So we have this flower, and the red petal phenotype it is coded for by the red allele, and the blue flower phenotype is coded for the blue allele. So I'm going to introduce the three different patterns of dominance, and they are complete of dominance, which you already heard of, codominance, and also incomplete dominance. I'm going to explain what these two new patterns are through this flower example. So let's start by looking at three different genotypes and the phenotype that you would see for each of them under each different dominance pattern. So we will start with the genotype through red R's, which we could expect that in all cases the flower petals will be red. Since we only have red R's in the genotype, similarly, if our genotype had two blue R's, then we could expect that in all cases the flower petals will be blue, since we only have blue R's in the genotype. These three different dominance patterns change when we look at the heterozygous example. That's what make these three patterns different. Now we're already familiar with the example of complete dominance. So if we said that the red R is dominant over the blue R, then this would make the heterozygous phenotype a red flower for complete dominance. Now, what codominance is? When the heterozygous phenotype show a flower with some red petals and some blue petals. So, in which the two alleles are dominant together, they are codominant, and traits of both alleles show up in the phenotype. Now, what incomplete dominance is when the heterozygous phenotype shows a mixture of the two alleles. So in this case, the red and blue flower petals we combine to form a purple flower. And now, let's talk about the allelic or non-epistemic interaction. So, epistemic gene interaction is categorized as follows based on how the evolved genes affect one another expressions. First is supplementary gene interactions, complementary gene interactions, inhibitory gene interactions, and lastly is the epistatis. In supplementary gene interaction, the phenotype effect is produced by the dominant allele of one or two genes regulating character. However, the dominant allele of another gene has no independent phenotype effect. So therefore, it changes the phenotype effect brought on by the first gene when it coexists with the dominant allele. For example, phagotype or Gray got development in mice. Complementary gene interaction. The F2 ratio becomes 9:7 instead of 9:3 if both gene loci are homozygous and reproduce the same phenotype. In this instance, a single phenotype is produced by the genotypes 2 small a and 2 small b, and 2 small a and 1b, b and small b, and 1 big A, small a and 2 big b, and 2 small a and 2 small b. When both dominant alleles are present simultaneously, so they are referred to as complementary genes and result in a distinct phenotype.